Again, this is Shawn Lee from Merck. The presentation will be sorry, have a little challenge to move forward yet. Here's the outline of the presentation. First, I'll provide a brief introduction of biopharmaceutical process development and host cell plotting. Later on, we are introducing two workflow for HCP identification, leverage the Evosep LC systems. So as you most of you know, right, for pharmaceutical going into patients usually goes through discovery, development, and manufacture. We know proteomics have been widely used in the discovery space to really identify drug target, elucidate the mechanism of action of those pharmaceuticals. But once the drug was discovered, right, the next question is how can we make it, and how can we ensure the quality? That's really exactly the scope of the development and the manufacturers. So here illustrated the typical process to make a biopharmaceutic, including cell line development, upstream cell culture, downstream purification, and also formulation development, right? So in this slide, we outlined several mass spec application to check the pharmaceutical quality, and also some process or excipient quality as well. But among all those mass spec applications, there was also proteomics opportunities, including using mass spec based multi-omics, including proteomics to check the cell growth, the cell growth status. And also another application which is the focus today is to check the process impurity, which is the host cell protein in both the upstream and the downstream space. So what is really host cell protein, right? I guess for this audience, don't need too much introduction. HCP are process-related impurities derived from host organism, include Chinese hamster ova cells or E. coli. So compared to the therapeutic protein maps, right? Those host cell protein are really undesigned proteins released by host cell by host cells. So why do we care about it? We know certain residual host cell proteins have potential, adverse effect for both safety and the efficacy of a bioproduct, including immunogenicity, acute toxicity, product and excipient stability. Here we list some, example of host cell protein, how this, adverse effect, including the HCPs, which is really identified and categorized by us, right? Including the protein called HEX-B and also a lipase called PLA2G7. This talk really focus on the excipient stability, right? So why do we care about? Formulation excipient such as polysorbate or called twin 20 or 80 really serve as a surfactants can protect the protein against interfaction stress in biopharmaceutical formulations. Enzymatic degradation by host cell polysorbate degradative enzyme, or we call the PSD here, such as lipases, is often considered as the dominant pathway for polysorbate degradation in biological formulations. Why do we care about it, right? So once those polysorbate was degraded, often they release the free fatty acid, which can form subviscous particles, which is a critical quality attributes we needed to control. That's why it's so important to make sure there was minimized polysorbate degradation to lead into subviscous particle formations. So how do we measure HCP expression and clearance for biopharmaceutical development? Right? The standard approach is ELISA, is relatively robust assay. Although it cannot have 100% coverage of the host cell protein, and it also can only provide a single measurement, and there are no identification and quantification of individual HCPs for process optimization and risk assessments. In contrast, LCMS-based proteomics has approved as a orthogonal analytical method for HCP characterization. It really provides identification and quantification of HCP for process optimization and risk assessment. So one of the downside is really, it's not really easy to implement for master qualification and testing in the GMP QC labs. I know the next speaker, Thomas, and his colleague at Aflysis have really did some, innovative work in this space, and really look forward to, Thomas' presentation, after these presentations. So this, proteomics workflow, right, by leverage different sample operation, used either in gel digestion or in solution digestion, coupled with analytical or nano LC, then we are be able to connect with the mass spec for, HCP identification and quantification. This really have been used as a platform approach for HCP characterization across different modality at Merck. At Merck, we have a rich pipeline across biologics, biocatalysts, driven the small molecule, synthesize, and also vaccines. And we have listed a number of papers by leveraging this platform approach for HCP characterization across all these modalities. We do see a few challenges, including low throughput. Often we run about 3 hours per sample, and is ready to poor assay precision for quantification, and sometimes lack the sensitivity to trace those high-rich host cell protein, often below sample ppm level. So the presentation here is really to address these challenges. So in order to address the challenge about throughput and robustness, I think we have been so fortunate to leverage the Evosep LC system. So in this case, for those people who are not really familiar with the Evosep LC, here shows a picture of the LC system. So one of the really interesting part of this instrument is they combining the sample clean up and the sample injection in the single tip. The process to prepare the tip is really simple, similar like what we prepare the zip tip for sample cleanup for either MODITOF or for ESR, LCMS analysis. So the advantage of this really provided to for HCP analysis across different sample and different modality is, for example, for high concentration HCP sample, such as for those in monoclonal antibody, essentially we don't have really limitation in terms of sample amount, right? We often use a limited digestion and then coupled with the LC, and we often offer overloaded the column, try to load as much sample as possible in order to identify this low abundance HCP. And during the limited digestion process, there was a centrifuge step to really remove the aggregates, but sometimes it's not effective. Sometimes during storage, new aggregates can formed. By using this Evosep system, we are really able to use the EVA tip to remove the aggregates, and we potentially can load less material, compared to a typical analytical, LC flow. In this way, we are able to really increase the throughput of the MAP HCP analysis. For those low concentrated sample, either for vaccines or for cell or gene therapies, often we leverage the nano LC system, right? And also for the work we demonstrate later on through the affinity purification approach for those low balance HCPs, we leverage the nano LC system. Often, the sample loading step takes a significant amount of time. By leveraging the Evosep system, we are able to really avoid the sample loading time from the trap to the analytic column. In this way, we are really able to increase the throughput and also the robustness, because the tip, often we just use once. So in the next 2 case study, I will really demonstrate how we leverage this Evosep to address both the throughput and the sensitivity issue, right? So the work is really done by a former postdoc in the lab, Taku. Right. In the first workflow, he developed a high throughput LCMS based proteomics workflow, leverages the Evosep coupled with the Bruker timsTOF. I should say, the Evosep can not only coupled with the Bruker instruments, it can be connected with other mass spec window as well. In fact, in the lab, we have been using Evosep LC coupled with the thermal exposed 480, and also recently we coupled with the thermal astral mass spec as well. So in this specific case study, Taku have been evaluating different approach to build the DIA library to for host cell protein identification. Follow the throughput side, right? Taku evaluate different sample injection from 50 sample per day to 300 sample per day. Essentially, we found the throughput by leverage the 200 to 300 sample per day. We have the identify more protein compared to the traditional sample we throughput we did about 8 sample per day, which significantly increased the throughput, but while we are still able to achieve significant amount of coverage in terms of host cell protein identification. For the precision side, right, we often also achieve a really good precision. We are able to achieve media CV about 60% compared to a traditional LCMS workflow set up in the lab, which is about 25%. Among different throughput methods. Of course, once the peptide have high abundances, we tend to see low CVs as expected. So how do we leverage this workflow to support process development, right? The typical or most used approach is really support downstream process optimization, which is the most widely used way to remove those host cell protein. But sometimes the downstream and toolbox could be exhausted, and we are still not able to mitigate all the impact for this high-risk host cell protein. Often we have to leverage our upstream cell culture to really reduce the loading amount or the initial amount of those high-risk host cell protein. So in this case study, which is published, in this publication shown here, we evaluated the one monoclonal antibody that was cultured under various cell culture condition and harvested at a different time point, and we also coupled with a lot of mass spec system solubility or sample blanks to really make sure the quality of data. We leverage 100 sample per day methods, and we are able to achieve really good reproducibility between between runs, and the media CV is about 6%, and we see virtually absence of carryovers. While we are also able to achieve close to 2,000 HCPs through this harvest cell culture sample, including over 30% of high-risk host cell proteins. Often the high throughput host cell protein workflow really work really well in terms of kind of screening tool to see what the high host cell protein really in the sample. But we sometimes still see challenges. So one is really on the sensitivity side. Some of the host cell protein, especially in their drug substance, they could be below ppm level, which is over 6 order of dynamic range. We know a lot of mass spec, really the typical dynamic range is 4 to 5 order, right? So another challenge is about the activity. We know those enzyme abundance and activity is not a linear relationship. So how can we solve those challenges, right? Are there any assay can really solve these two challenges, in a single assay? So the answer is yes. So when we did the little research, we found that those polysorbates enzyme or PSDs, such as lapises, that belong to a serine hydrolysis superfamily. So one for the signature of this family is they have a neutrophilic serine in their active site. So this really shows the mechanism action of the serine hydrolysis catalyze. So based on the mechanism action, there was a lot of research done in the academia, especially the work by Professor Ben Kravitz for Scripps. They developed a lot of this active site direct activity-based probe, such as the FP biotin shown in this case. By leveraging this chemical probe, we have been able to specifically label those active enzymes. Then we can use the evidence beads to specifically enrich those labeled enzymes. So in this way, we are able to significantly remove those sample matrix. For example, monoclonal antibody in this case. So compared to a traditional workflow, we are able to significantly reduce the background, increase the sample dynamic range, and also while provide the activity readout. So in this case study, there was two map. One of them have significant polysorbate degradation through the incubation study. And if we run abundance proteomics, we did not identify really any HCPs. And if we run the activity-based proteomics ABPP, we are able to identify two lapases or esterases in this case. While for another map, we did not see significant degradation, and we only identify PLBL2 by abundance proteomics. We know PLBL2 is not responsible for polysorbate degradation, and if we run activity-based proteomics, we did not identify any active lapases in this sample. So really, in order to confirm the PLA2G7 really have an impact on polysorbate degradation, we did sparking experiments. We see PLA2G7 was able to both time and dose dependently degrade polysorbate. In this case, PLBL2 was served as a negative control. LPLA2 was served as a positive control. So really, by leveraging the initial workflow, we still sometimes show challenges to identify those low abundance enzymes. So Taku really, leverage the Evosep system, did a systematic optimization of the ABPP to really identify what step can potentially even increase the sensitivity of the ABPP. Through the DOE design, Taku found the FP biotin concentration, and also the initial pH condition for the incubation during the incubation step, really leading to most significantly improve the sensitivity of the ABPP. As I mentioned before, this work is really through the significant DOE design. We have a lot of sample to be tested. In this case, the Evosep really helped a lot in terms of throughput. This shows the throughput of the ABPP workflow, right? Essentially, we run 60 samples per day, can really achieve similar sensitivity compared to the conventional nano LC system we used in the past, about really only run like 8 samples per day. So we also evaluate different, mass spec acquisitions, including TDA PASAF, DIA PASAF, and also PIM PASAF. By using two sparking enzyme, PLA2G7 and also SIAE, we found the optimized workflow for ABPP can identify those enzymes down to 10 ppb level. For certain enzymes, such as the SIAE, we can also confidently identify them in the 1 ppb level. This is really the lowest that have been reported so far in the literature. So why is this important, right? So in this case, in one of the map, using the UFP sample, using the original ABPP sample, workflow, we loading more than 20 milligram. We only able to identify one lapacid. And we know this was not the leading cause of polysorbate 80 degradation. If we run the new workflow, loading much less material, we will be able to identify one additional, hydrolase, which might be the leading cause for the polysorbate degradation. We will be able to feed this information to our process team. They are able to optimize the process to really in order to remove the polysorbate degradation issue to ensure the product quality. So this was really the summary of my presentation. I hope I will convince you the LCMS-based proteomics have really great potential in biopharmaceutical process development and manufacture, and advanced in LC and also mass spec for high throughput proteomics and ABPP really enable the robust process development and risk assessments for the control of high risk host cell protein. This really the work showed here really is a true collaboration across, analytical process and the formulation department within Merck. Specifically, I would like to thank the postdoc Taku really drive the innovation to help us reach the data which we showed in this presentation. But we also wanted to thank the mass spec team within Merck, and also the polysorbate education and the lapis control strategy working group at Merck. We have been working in these industry challenges probably over 10 plus years to really drive the innovation in this field to make sure we really develop the high quality pharmaceuticals to the patients we serve. With that, thank you for your attention, and happy to address any questions at the end of this webinars.